Hello everybody, my name is Emmelien Vancaester and I'm a postdoc in the lab of Professor Wolbachia and I'm part of the Darwin Tree of Life initiative. I want to thank the organizing committee for giving me the opportunity to present my research here today. I want to talk about Wolbachia, which is a common endosymbiont sequenced within the insect samples as part of the Darwin Tree of Life project. So Wolbachia is a common intracellular alphaproteobacterium which can infect arthropods and nematodes. Estimates on occurrence of Wolbachia widely vary. It is estimated that up to half of all terrestrial arthropods and even 80% of all Lepidoptera species can be infected by Wolbachia. Interactions with its host are often complex, ranging from mutualistic to parasitic. While it has been shown that some species display high fitness levels compared to other uninfected members of its population, Wolbachia can also have a detrimental effect on its host. Wolbachia is maternally inherited in its host and typically infects the gonad tissue. It often interferes with sexual differentiation of its host and can do so in 4 different manners, which are male killing, feminization, induction of pathogenesis, and cytoplasmic incompatibility, with the aim of increasing its spread throughout the population, while simultaneously causing sexual imbalance. Species in the Darwin Tree of Life framework are sequenced using the PacBio-SQL2 HiFi platform, which generates highly accurate long reads, which are on average 10 to 15 kilobases long. This allows to determine the species composition of each sample, as some will contain parasites, endosymbionts, and contaminating DNA, etc., by searching for a small subunit ribosomal RNA. For Wolbachia-positive samples, the long reads were classified using Kraken based on a database reflecting composition of the sample to extract the Wolbachia reads. These were then reassembled with both HiFathom and Fly, choosing the assembly with the best completeness and contiguity. So we screened 368 insect genomes for the presence of the intracellular endosymbiont Wolbachia. Wolbachia's small subunit RNA sequences were then detected in 111 or 30% of the species. Incidence was slightly lower in Coleoptera or the beetles compared to Lepidoptera, Diptera, and Hymenoptera. However, Wolbachia prevalence and infection intensity varies between species and between populations within a species. As only one individual was analyzed for each of the insect screens, the true level of infection within the insect biota is likely much higher. So in total, we generated 110 complete genomes, of which 77 were circular. We even detected 15 species which were co-infected with 2 or more Wolbachia. These assembled genomes are of high quality, as shown in the figure here, with the novel genomes assembled in at most 6 contexts. So on the left-hand side, you can see the contiguity of these genomes on the upper side, while on the lower fraction of the slides, you can see the reference genomes which are currently present in the databases. On the right-hand side, you can see their assembled genome size, which is on average higher compared to the reference, probably because the long reads allow to assemble across repeated loci. So Wolbachia is a genus consisting of at least 17 supergroups, of which you can see a schematic representation on the right-hand side. Most sequences deposited in databases today belong to either supergroup A and B, which are also the groups which infect insects, with supergroup F being a group that can infect both nematodes and arthropods. So adding those 110 newly assembled genomes to the reference genomes yielded a dataset of over 200 high-quality assemblies. We annotated all genomes using Protocol and clustered the predicted protein sets into orthologous groups using OrthoFinder. 634 near single copy genes were used to infer a phylogeny of Wolbachia. We classified all our newly generated genomes by their supergroups, and they either belong to supergroup A and B, and increased the phylogenetic diversity of these groups. We can also compare the phylogeny between the insects, here shown on the left-hand side, with Wolbachia, here on the right, by drawing a gray line between each of the Wolbachia strains and its infected hosts. Wolbachia generally do not show strict cophylogeny with their host, as seen by the many tangled lines. Closely related insect species may be infected by dissimilar Wolbachia strains, and conversely, closely related Wolbachia can infect a diverse set of insects. However, Lepidoptera were predominantly infected with supergroup B, while Wolbachia supergroup A was most frequently observed in all the other insect orders. We can also have a look at the gene content. An interesting example is biotin, also known as vitamin B7, which is an essential metabolite that needs to be taken up in a diet by most insects. Interestingly, the operon-producing biton was detected on only 14 genomes across the whole dataset of 203, of which 7 are novel, indicated in bold. The Wolbachia harboring this locus show a punctuated pattern seen by the dead rods in the phylogeny here on the right. The operon is strongly conserved in its physical organization of all 6 necessary genes, which is shown centrally in black, in a black box. In the genomic region immediately surrounding this operon, we identified many recombinases and transposases. And in even 3 of the genomes, the operon was independently disrupted by transposases, as you can see here, over here, and here. Moreover, we could also detect anchor and repeat containing genes and a toxin-antitoxin pair of cytoplasmic incompatibility genes close next to this biton locus. So this region containing the biton operon has the hallmarks of being a so-called virulence island that may be mobile between genomes and may have accrued additional genes that can hitchhike along the biton operon, which might confer a positive advantage. Moreover, 8 of the genomes which contain this biton cluster are from Wolbachia infecting bees, namely bees belonging to the genera Lossiglossum, Andrena, and Nomada. This distribution suggests there might be ecological linkage, as Andrena bees are kleptoparasitized by Nomada cuckoo bees, and the phylogenetic analysis of both the biton cluster as well as the core proteome of the Wolbachia show very close relationship between those bees. Wolbachia is infecting the bees. Wolbachia itself can also be infected, this by a double-stranded DNA temperate bacteriophage, the W-phage, which can integrate in the Wolbachia genome as a prophage. These regions were identified, and the fraction of the total prophage region in each genome ranged between 0 and 30%. Nematode-associated Wolbachia cannot be infected by the W-phage, and no prophage regions were detected. A significant correlation was found between genotypes and W-prophage span in supergroups A and B. This association was robust for correction for phylogenetic relationships of the genomes. The largest circular genome assembled to date, which was generated in this study, a Wolbachia infecting the blue-tailed damselfly Ischnure elegans, also had the largest prophage span. So in conclusion, the data generated by the Darwin Tree of Life doesn't only result in chromosomally resolved genomes of the host, but also creates high-quality symbiont genomes. Lepidoptera are mainly infected by supergroup B and other insect orders by supergroup A. However, host switching events are very rampant in the evolutionary history of Wolbachia. And finally, Wolbachia genome size is strongly influenced by W-phage integration. So I want to thank everybody from the TTOL team for making this work possible, from the collectors who obtained the specimens, the extraction lab, the sequencing teams, and the curators. I want to thank Mark Blackster for his continued support and the whole team for input during the whole duration of this project. And of course, thank you for listening and taking interest in my work. Thank you.